All right, today we are going to talk about two really important topics when we're talking about evolution, and that is the concepts of populations and species. So the definitions of what are a population and what is a species are really important, but they also grade together into one another. So I think it's important to talk about them together, but this material is covered in two different chapters in your textbook. So we're going to look at the very smallest amount of evolution that can happen. And so we're going to call it microevolution, like a microscope. So microevolution is the smallest amount of genetic change that can be detected in a population. And we're going to refer to that as a change in allele frequency. So if you don't remember what an allele is from learning about about Mendelian genetics, we'll re-explain that a little bit. And then we're also going to talk about what is a species, why it's important to keep them separate from one another, how do we get a new one, and what happens if they don't. So lots of interesting topics today. So when we start looking at population genetics, that is, what is the genetic information that is together in a population, it turns out that's how we're going to be able to look for evolution happening. We don't look for evolution happening in one individual at a time. We only look for evolution happening to the population as a whole. So we're going to see if there's new genetic information that's coming in, genetic information that's getting lost, changes in proportion of how many individuals have each allele. And it turns out there's 4 ways that those allele frequencies can change. So we'll talk about natural selection, genetic drift, mutation, and migration, right? So these are the 4 ways that evolution can happen. If you've already learned about natural selection in another class, then we want to make sure that you understand that there's 3 other ways that allele frequencies can change, and they cause changes to the population in different ways. Our second topic is speciation. How do we keep species apart from one another once they are formed? Because that's important to understand before you can understand how do we get a new species in the first place. So speciation is the event that happens where we had one species and it turns into 2. So if we were drawing a cladogram, then we would show that node, right? So we had one species down here, and then some time passes, and now we have 2 species, species 1 and species 2. Now, usually species 1 and species 2 are going to be different from the ancestor we started out with. Sometimes one species will stay pretty much the same, and then a second one will branch off and become different. So this could be how the speciation happens. So this number 1 down here means that it could be very similar to the species, one of the new species that we get. All right. So if we have 2 species that are distinct from one another, what's a population? Well, populations are subgroups within a species. So if we imagine that this species 1 occurs over a geographic distribution, it doesn't want to draw, right? So species 1 occurs across a geographic distribution, but that there may be some places where they don't occur. So this is species 1, population A, and population B, right? So we're going to find, a large number of individuals in each of these regions. And these are both in normal habitat area for species 1. In order for them to stay the same species, there has to be some genetic interchange between them. Right, so a few individuals are going to move in between areas A and B. So if we're talking about birds, one will fly from one place to another. If they're completely well mixed, we're going to say that they're all one population. But if there's some separation between them, we're going to say they're two populations. If they're completely cut off from one another, there's no mixing, no migration, then although they may be the same species right now, they're probably not going to stay the same species into the future. So that's a way that species can occur, is that we have two populations that are cut off from one another, and they just start evolving separately in two different directions. Right. So most of the time, though, most species exist in this spatially structured way, that there are more than one somewhat separate groups from one another. We call those populations. Populations may be quite well mixed with one another, or they may be very little mixed with one another. So there's a broad continuum, right? So when we say that this and such a thing is defined as a species and this and such a thing is defined as a population, it turns out that there's a wide variety of degrees of apartness that these groups can have. And somewhere along this continuum, we want to arbitrarily cut off and say, okay, these are two different species from one another. But it turns out that in nature, it may not be so easy to make that cutoff. So we want to start. So that's sort of the take-home message from the entire lecture, is there's this gradation between populations being very similar to one another, to very different to one another, to so different that we want to call them separate species. All right, so first we want to define a species, and we want to define a population. So let's get some formal vocabulary. This is an example for a population, and it's of caribou or reindeer. And the map shows where these two different herds. So they form herds or groups of many individuals that have a social behavior where they stay together. And in Alaska and the Yukon Territory of Canada, they are going to range, or that is roam, over these large geographic areas. But they're each of these two herds, the porcupine herd and the 40-mile herd, they're going to stay in their own distinct areas. Now, although they overlap geographically, they don't use those areas at the same time, right? So this is a way that by behavior, these different groups are going to stay for the most part separate from one another. So when we have a behavior like this herding behavior, we can define each of the herds as a population. All right, so a population is going to be a two or more groups. We can have more than two populations, a group of individuals of the same species. So the things inside a population are all the same species as one another. So a group of individuals of the same species that are somewhat isolated from other such groups, right? So that definition can be a little hard to understand exactly what it means when it's written in a very simple form like that. But we're going to keep talking about it as we go throughout the lecture so that it completely makes sense in case it doesn't quite yet. A species, on the other hand, and I don't have a figure to show this, but a species we're going to define, and we're going to use in this class the biological species definition. Right? So there's different ways that we can define species. The best way is to go out in nature and look and see how organisms choose themselves to stay in a species or separate themselves from other individuals. But if we want to have a situation where we can have a good definition of a species, it turns out it's a little difficult. And so there's actually different scientists who have tried to write better or different definitions of what a species is. So there's a whole long list of different ways that we can define a species. When we first started being able to do rapid large-scale DNA sequence analysis, we first thought, the scientific establishment first thought, oh good, now we'll finally have an arbitrary, objective way to tell when two populations are different enough to be a species. We'll just look at the DNA similarity and we'll get a certain percent difference in DNA and we'll be able to say, well, above 10% difference in DNA or whatever it is we're looking at, we'll be able to say those are two different species. And if it's less than that, then they're the same species. Well, it turns out it's not that simple because different species have different amounts of genetic information, genetic variability to start with. So there wouldn't be one hard and fast percent that we could use for that. And also when we look at different kinds of genetic information, whether we're looking at mitochondrial DNA or whether we're looking at nuclear genes, when we look at different genes, we find there's different levels of genetic difference between different species. So it turns out that isn't the simple answer either. All right. So most of the time we can use this biological species concept or the BSC. So a species concept is a set of rules that we're going to use to decide if two individuals belong to the same species or not, right? So a species concept is a set of rules that we're going to use to decide if two individuals belong to the same species. The biological species concept says that we're going to do this by taking two individuals and seeing if they will mate, can mate with one another, want to mate with one another, and they'll be able to produce viable, fertile offspring. So it's a little tricky, say if we have two female individuals and we want to compare them, or two male individuals and we want to compare them, because they can't mate with one another anyway. So it's a little biased towards sexual reproduction. But in general, the results, if they do mate with one another, is what we're going to use to tell if they are a separate species from one another. If they will mate, but they don't produce any offspring, then they're different species from one another. If they do mate and produce an offspring, but that offspring is infertile, it can't reproduce itself, then we'll say they are not the same species. So we'll look at some more examples when we get further through the lecture. And at each of these different steps, we'll see that there are ways that different species have to enforce this rule to separate individuals from one another. When we think about what a species is, it is a unified group of chromosomes that have genetic information on them that have co-evolved. So all the genes that are in that genome have to work together to produce an organism that is adapted to its environment, that fits with the characteristics of the environment. So it's important that the genetic information that makes up that species stays intact. It doesn't get mixed or diluted or watered down with the genetic information of another species that may not work together with the information that that species has for itself. All right, so that there is a strong evolutionary need for species to have integrity. That is, that they don't mix with other species. However, sometimes it happens that two individuals of different species do mate and produce viable offspring. In that case, we'll say that offspring is a hybrid, right? A hybrid, H-Y-B-R-I-D, like a hybrid car. So even though we have these rules of what should happen, in nature, it doesn't always work that way. It turns out there are, in fact, some species that regularly hybridize with one another. And It may be an advantage, it may be a disadvantage, but it's actually much more common than we used to think, right? So we as scientists, we as people may want to put a hard and fast set of rules onto organisms and say, well, this is a species and this is how it should behave. It turns out that in nature, things are much more complex and varied than that, right? So your Bio 240 understanding of what a species is should start to include some of this sort of nuance and gray area, okay? All right, hopefully you think it's interesting and exciting and not just overwhelming and confusing. All right. Excuse me. So if we're going to start to think about how populations might differ from one another, we need to define this concept of an allele frequency. So remember that when we can have different versions of a gene, so we have a gene for eye color, say, every adult human has 2 copies of these genes because we're a diploid organism. We could have the same exact genetic sequence on each of those 2 homologous chromosomes. In that case, we would call each of those individual sequences, the one that makes blue eyes, the one that makes brown eyes, the one that makes green eyes, we call each of those different ones an allele. All right, so an allele is just a different version of a gene. It's going to make the same thing. It's going to get used in the same way in the body, but it's going to be a slightly different version of it. So the easiest examples to think of are ones where there's a color. So flower color, we might have a purple or a white flower. Those are different alleles. One of the other things that there may be lots of different alleles for is lots of different kinds of proteins in the body. And a lot of proteins are enzymes, right? So we might have different versions of that enzyme. Now they may all work. They may work slightly differently under different circumstances. So when we're looking at marine organisms, one of the things that's important to them is the ability to do homeostasis, to regulate their body water. And when they're in an environment that is semi-marine, semi-freshwater, and they have changing amounts of salinity throughout the day, they may need different versions of some enzymes that have to do with how they regulate their water balance in order to survive better in the different salinity conditions. All right, so we have an example where we're looking at an estuary. An estuary is a semi-enclosed body of water that has a river leading into it from one end and is open to the ocean at the other. So it's got fresh water coming into it, depending on the time of the year and how much rainfall there is. There may be lots of fresh water or there may be a little fresh water. And at the other end, it's the open ocean. And so there's a salinity of seawater. As the tides rise and fall, there's more higher salinity as that seawater pushes the fresh water back up into the estuary. And then as the tide goes out, it'll have more fresh water. So we see organisms that live in estuaries having stressful environmental conditions because they're changing. It's easier to be adapted to a stable condition than it is one that's changing. All right, so suppose we know that there's an allele that is used for related to osmoregulation, that's regulation of body water and salts, in these blue mussels, which is a kind of shellfish, a bivalve. They're blue or sort of purple, little ridges on them. All right, so we've looked at populations of these mussels across a transect or a gradient, starting with this level, station 1, that's further up on the freshwater side. And then as we go out to more salinity, as we go across 2, 3, 4, et cetera, and all the way out to full seawater at 10. So we looked at the frequency in these populations. So mussels were sampled, their DNA was extracted, and some kind of analysis was done to look at what alleles they had. It turns out this LAP94 allele, which does imply that there's at least 94 different versions of this gene, this LAP94 allele has something to do with how they regulate their body water. So we looked at that one versus all the other ones. And the little pie charts across show these allele frequencies. So in the population at station 1, we see there's a little narrow pie wedge for the LAP94 allele, but that the population is dominated by these other LAP alleles. As we go towards the higher salinity sites, we see that steadily increasing proportion of the population has this LAP94 allele. So we can infer from these data that the LAP94 allele does better. It helps survival and reproduction in organisms in the higher salinity environments. Right? So it has a higher proportion. So when we're looking at populations and we say, okay, they're somewhat isolated from one another, we may see that they have somewhat different genetic characteristics. Now, we aren't going to see that the muscles look different or that they aren't going to mate with one another. We just happen to find that in some populations that they have a somewhat different proportion of certain alleles. Certain alleles are a little better in that environment. So we can have populations that are somewhat different from one another. They can be locally adapted. And this change in allele frequency from one geographic area to another, we call that, it's evolution, right? It's a change in gene allele frequency, right? So this change in allele frequency that we can detect is our smallest amount of evolution that we're going to be able to see. So we call it microevolution, right? A change in allele frequency. Now remember that each individual, if we're looking at adult animals, has 2 copies of each gene. So they can have 2 alleles. So if we counted 50 muscles, then we would say out of those 50 muscles, there was a total of 100 alleles represented, right? Each individual could have 1 copy of the LAP94 allele, or it could have 2 copies of the LAP94 allele, or it could have 0 copies of the LAP94 allele. So we count up how many total LAP94 alleles there are out of those 50 individuals, and then we divide by 100 because that's how many alleles they have, okay? I'm going to do some worked out examples of the math for all of these situations in our in-class version of this class meeting. So if you're not quite following along on the math, you can review it in the textbook, and I'll cover it when we are talking in person. Math is very easy. All right. So before we start talking about how we can detect a change in these allele frequencies, we have to do a little bit of Mendelian genetics. So I think this cartoon is funny because Gregor Mendel was a monk and he did all of his experiments on pea plants. So you can imagine that the garden in the monks, the place where the monks live, was making an awful lot of peas compared to other kinds of foods. And so the other monks are sitting around the table and he's bringing them peas to eat again. And they say, we're tired of peas. All right. So basic Mendelian genetics, we need to do this to understand population genetics. And I'll also have linked a more detailed review of Mendelian genetics for you. So if you've heard this before, a quick review is going to help. If you never learned it at all, you're going to need a little more time. All right. So Gregor Mendel started with true breeding strains, which meant that his red flowered pea plants and his white flowered pea plants, every time he bred a white with a white, he got only white offspring. And every time he bred a red with a red, he only got red offspring. It turns out that means that those individuals were all homozygous. That means they had the same allele on both of their copies of their chromosomes, right? So calling the red allele big R, the red flowered plants are big R, big R, the white flowered plants are little r, little r. That combination of those 2 letters, big R, for example, we're going to call it the genotype. So whatever code we use to write down the different alleles, if we're talking about a diploid organism, we write down those two alleles that that individual has. That's the genotype. Now the phenotype, spelled with a PH, phenotype is what observable characteristic does that produce in the individual. So the big R genotype produces a red flowered phenotype, and the little r genotype produces a white flowered phenotype. Now, when those plants go through meiosis, they are going to make haploid gametes. So flowers indeed produce sperm and eggs, just like animals do, in that they make sperm and eggs. So each of those gametes is going to be one of the alleles that the parent has. The big R individual can only make big R gametes. The little r individual can only make little r gametes. So if we cross together the red flowered and the white flowered, in this example, we get all pink flowered plants. Okay. So when we think about what genotype, how the genotype is going to cause the phenotype, all of our offspring are going to be big R, little r, because one parent gave a big R, the other gave a little r. So those are going to be called heterozygote, meaning mixed or different, so big R, little r. Now, you may have read about purple versus white pea plants from Gregor Mendel's work. It turns out that the way that the color works in the purple versus white gene is different than how this gene works in this plant. So in the purple versus white flowers, the heterozygotes are purple. So that big P purple allele exerts complete dominance over the little p white allele. The reason for that is that the little p white allele doesn't actually make a white pigment. It actually makes no pigment, right? So if there's any purple pigment being made at all, the flower is going to look purple, right? So that whether you have one big P allele and one little p allele, or you have two big P alleles, purple pigment is being made by that one side of the DNA. Purple pigment is going to be filling up the cells of the petals, and they will look purple. So there's complete dominance. This example is an example of incomplete dominance, and that means that instead of completely hiding the recessive or little r phenotype, we get a mixture. We get some kind of intermediate phenotype. This is because in this case, in this plant, the little r allele, it doesn't make no pigment or a pigment that doesn't make any color. It makes a pigment that actually is a white pigment, right? So just like when you are buying white paint or white nail polish, there actually it's not clear. It actually has to have molecules in there that absorb all wavelengths of light so that it looks white. It doesn't reflect any colors back to your eye. So imagine that the white allele is like white paint. So we mix it with the red paint, and we get pink. So this example of incomplete dominance shows us that the phenotype of the heterozygote is going to depend on what each of those alleles DNA sequence makes as its protein and how it interacts with the other alleles, right? So you can't necessarily know what that heterozygote is going to look like unless you know how the gene works, all right? So here's our example of incomplete dominance. All of our pink flowered plants are big R, little r. The gametes that they make are going to be 1/2 big R's and 1/2 little r's, because during meiosis, all the big R's and the little r's are going to separate from one another. As a result, we will get, and we're summarizing what we get in this little 4-box diagram called a Punnett square, Punnett. And we're going to put the possible sperm that the dad can make across the top of the box. We're going to put the possible eggs that the mom is going to make. Each of them is going to have 1/2 big R's, 1/2 little r's. And then we're going to fill into the boxes the offspring that will be made if this allele from the dad combines with that allele from the mom. All right, so if we get a big R sperm and a big R egg, we're going to get a big R, pink flower plant. If we get a little r from the dad and a big R from the mom, we're going to get a little r, big R. If we do the opposite and we get big R from dad and little r from mom, we're still going to have a pink flowered heterozygote. And then if we get little r from dad and little r from mom, we get the white flowered offspring. So these are going to be the predicted possible kinds of offspring that we can get from this combination. And we can also find the probability, the predicted probability of offspring of each of these different kinds. If we have many plants and they're making many offspring, thousands of offspring, even hundreds of offspring would work, we can use the mathematics of probability to predict what proportion of offspring we would get. Mathematics of probability work like this. Whatever proportion or probability we have of getting a big R sperm, we're going to take that event and we're going to say, what's the probability of getting this event and this other thing coming together and happening at the same time? We're going to multiply those probabilities together. So we're going to take 1/2 times 1/2 and we're going to get 1/4. So each of the cells, each of the 4 cells in this Punnett square has a probability of that thing happening of 1/4 because we're looking at meiosis happening at the top. And so exactly half of the sperm are big R's and exactly half of the sperm are little r's. If we want to count up the total number of heterozygotes, then we say, well, we can get a heterozygote with the big R from the mom, or we can get a heterozygote with a big R from the dad. You can have that 2 ways. So we can get them this way or the other way. So we first calculate the probabilities of each of those ways, which are 1/4 each. And then we add those together to get the total frequency of heterozygotes. Right? So we're going to add together 1/4 and 1/4 and get 1/2. Now, your instructor might have told you when we're doing Punnett squares for Mendelian genetics, we can look at how many squares there are in the table here, and we can count up the squares, right? So instead of some complicated probability math, we could just say, well, there's 1 square here out of 4 that's red, so we have 1/4 probability of getting big R's. That works for Mendelian genetics because each of these combinations of alleles has the same probability and it's exactly half. We'll see in a minute that we can't do this when we're doing population genetics because these numbers won't be exactly half most of the time. All right, so you can only count up the boxes to find your proportions if you have the same proportion of the big R and the little r alleles. Okay, so if instead of looking at what happens if we have one specific parent with a known genotype and another specific parent with a known genotype and what offspring can they produce, that's Mendelian genetics, we're going to switch gears and talk about population genetics, right? Population genetics is basically we take all the adults in the population, we shake out all of their gametes, we look at what alleles they have, and we look at the proportion from all the individuals taken together. Right? That's what we were doing when we were looking at the example of the blue mussels, looking at the proportion of the LAP94 allele in the population. So the mathematics of population genetics is based on the assumption that we are taking all of the adults and shaking them out, which doesn't seem very similar to, say, how humans go about choosing mates and mating with one another. But if we have a very, very large population, these assumptions work pretty well. The organisms like blue mussels, which release their sperm and eggs into the water and then everything just mixes together, those kinds of organisms match very well to this assumption or this prediction. All right, so we want to find the frequencies of the alleles. Remember, each individual has 2 alleles. So first we need to find the genotype and then count up all the alleles. We're going to find the frequency of each of the alleles. So in this population, we found that there is this red allele and it has a 0.8 probability or a proportion, and we only have 2 alleles. So the sum of the 2 allele frequencies has to add up to 1.0. So the frequency of the white allele equals 0.2. So we're going to assume when we're making a Punnett square that there's all these possible combinations of alleles, just like we did for Mendelian genetics, but that instead of having a probability of 1/2, they have whatever probability we have found is present in this population. So each egg, instead of having 50% probability of the red allele, it's going to have 80%. All right. So we need to know those allele frequencies in the population to start with. What we're going to do is exactly the same math as we did for the Mendelian genetics, except instead of starting with 1/2 and 1/2, we're starting with 0.8 and 0.2. So we're going to put together the red and the white alleles. And in this case, the notation's a little different, right? We aren't just using a big R and a little r. We're using this capital C and then a superscript big R for the red allele and a superscript big W for the white allele. So there's lots of ways that we can write notation for our alleles. And in fact, in many species, there's many alleles. There's much more than 2. So we can't just use big A, little a, right? So like the LAP allele, that LAP is the name of the enzyme, and then each different allele just has a different number. So there could be a LAP 5, there could be a LAP 7, right? So you want to become comfortable with using notation to denote different alleles that is not just big A, little a, okay? All right. So if we have an 80% or 0.8 proportion of the big R allele, then to find the proportion expected in the offspring of the whole population taken together, not just of one parent, but of the whole population taken together, we're going to take 0.8 and multiply times 0.8 and get 0.64, right? So that's not 1/4 because we have a predominance of these alleles in the population. We're going to do the same thing for the heterozygotes. We're going to take 0.2 and multiply times 0.8 and get 0.16. Right? Remember, when you're converting from percentages, that you're going to move the decimal place. If this is 20%, we're going to divide by 100 to turn it into a decimal. You don't want to multiply percentages together. You want to multiply the decimal values. Right? So I don't like in this example that it's used percentages because we're going to do the 0.2 times 0.8. We're going to get 0.16. Same thing for the other heterozygote. And then for the homozygous recessive, we're going to do 0.2 times 0.2, and that gives us 0.04. All right, so it's drawn these boxes not even to one another to show that the proportions are different, if that helps you understand what we're doing here. So our total expected percent of heterozygote offspring, we still need to add these 2 boxes together. So we're going to get 0.16 plus 0.16, which is going to equal 0.32. So we're going to find probability of getting a heterozygote, CWCR, is going to be 0.16 plus 0.16. All right. Now the sum of our three different genotypes should add up to 1. So 0.64 plus 0.32 plus 0.04 should add up to 1.0. All right, so there's a little notation I want to make sure that you're noticing here. So I'm going to go back to the last slide. And over here it said, let's call little p the frequency of one of our alleles. And let's use little q to denote the frequency of our other allele. If we only have two alleles, then little p plus little q should equal 1.0, all right? So if we use little p and little q, what we're doing when we do these multiplications is we're doing p squared to find our heterozygous dominant frequency of offspring. We're doing q squared to find our homozygous recessive, and we're using p times q or q times p. We can write it either way. Multiplication is associative, and we're going to take 2 times that. All right, so the frequency of our heterozygotes is, I'm going to write it right down here. We can simplify it by saying 2pq. All right, so once we have the allele frequencies, we don't have to draw out the whole Punnett square. We can say, well, the proportion of big R alleles is going to be p squared, and I know little p, so I can square that. Right, I told you the math wasn't going to be hard. Little p times little p. Right. Similarly, we can find the expected proportion of heterozygotes with 2pq and the expected proportion of homozygous recessive as q squared. All right. So these are our expected proportions, assuming nothing happens except random mating, a large number of individuals, and the important characteristic that no evolution is happening. All right, so this is saying that if nothing interesting is happening in this population, then what I expect to find due to randomness and the rules of probability is going to be based on these proportions, p squared, 2pq, and q squared. So this doesn't seem very exciting, except that it gives us a way to test and see if evolution is happening, right? This is an earth-shattering revelation, people. This gives us a way to test and see if our genotype frequencies changed from one generation to the next, right? We can also test to see if our allele frequencies change from one generation to the next. We aren't going to do some real sophisticated math. We aren't going to do a hypothesis test. We'll do that in a statistics class. You might do it in the upper division genetics class. We're not going to do it here. But the idea is, if nothing is happening, we should have offspring that match these proportions. If something's happening, we'll have offspring that don't match these proportions. There's a couple ways that, a couple things that could be happening that would make our proportions change from one generation to the next. All right, so we're going to assume that our population is in equilibrium, right? The population's in equilibrium from a population genetics perspective if its genotype frequencies are not changing from one generation to the next, right? All equilibria have to do with something not changing from one time to the next. In this case, it is going to be the genotype frequencies. We're going to call this equilibrium Hardy-Weinberg equilibrium because those are the scientists that first wrote a paper about it, right? So this idea of genotype frequencies not changing from one generation to the next is called the Hardy-Weinberg equilibrium. The Hardy-Weinberg equilibrium proposes that if we have parents that have allele frequencies little p and little q, that their offspring should have the expected proportions that we just derived here, p squared, 2pq, and q squared. So what we're going to do is we're going to calculate those expected proportions, and we're going to compare them to what we see in the population. If those two sets of proportions differ from one another, then that's evidence that evolution could be happening. Right now, we also have assumptions that sperm and eggs are coming together at random. So assuming that organisms are having some kind of mate choice and they're not coming together at random, that would be called non-random mating. So it's possible non-random mating can be happening. It's also possible that if we have a very small population or a small sample size, that our predictions are not going to match to what we would expect to see based on the rules of probability. The rules of probability only work if we have a large enough sample size that we see all of the possible outcomes in the expected proportions. That's a definition of the probability. So if we have a small sample size, then we may not see those proportions either. So if our population is not in Hardy-Weinberg equilibrium, it could be a small population and we have a small sample. They could be non-randomly mating, or evolution is happening. Okay, so we'll see. The next couple slides are about what are the different ways that evolution could be happening. I have some problems I'd like you to be able to work out using these calculations. I will walk you through all those steps when we're meeting one-on-one. And you'll have a homework assignment that asks you to practice. All right, so the first way that evolution can happen or allele frequencies can change over time is basically the same as this idea that if we have too small a sample size or too small a population, that random changes can happen just because we aren't going to match the rules of probability. If you flip a coin 10 times, it's possible you could flip it 10 times in a row, you could get ahead. Doesn't mean there's nothing wrong with the coin. It doesn't mean you didn't flip it right. That's how probability works, right? If you flipped it 10,000 times, you should get really close to the true frequency. But if you only flip it 5 or 10 times, you may get something that's very different to what we would think of as the true proportion, right? So this example is showing one of the ways that allele frequencies can change, and it's called genetic drift. Genetic drift, as in the allele frequency is going to drift from one side to the other. It's not getting pushed purposefully. It's just going with the flow. It's going one direction, basically just because of chance or bad luck. So if we imagine a population that starts out with proportions of little p of 0.7 and little q of 0.3, we see the flowers that are shown in that left panel in generation 1. The ones in the white boxes are, for whatever reason, just bad luck. Those are the only ones that reproduced, right? So what ends up happening is if only those individuals reproduce, we look at the next generation and we see that there's fewer red flowered plants just because the wind blew the wrong way, right? So in one generation, because those red flowered plants didn't reproduce as much, not because they were worse for environmental conditions, but just through bad luck, we can have a big change in allele frequency. We went from 0.7 and 0.3 to 0.5 and 0.5. Right, same thing happens, only in this case, the wind blew and only the red flowered plants happen to reproduce successfully for whatever reason, right? And then in the next generation, we end up with only red flowered plants. So we have little p equals 1.0 and little q equals 0.0. So in this case, this allele has become fixed in the population. We have reduced genetic variation. There used to be 2 alleles and now there's only 1, right? This happened just due to random chance. All right, so if we have a very small population, these kinds of genetic changes can occur quite rapidly over several generations, and they're just due to bad luck. All right, it's still evolution. It's still evolution because we're having a change in allele frequencies. It's not beneficial to lose genetic variation. So most of the time, genetic drift is considered a negative, right? It's going to cause some harm to the population. People are interested in conservation biology, people who are doing captive breeding programs in zoos. We have a small population size. Those zookeepers are very concerned about arranging the matings between the few individuals we have in captivity to try and minimize inbreeding, maximize the amount of genetic variability among individuals so we don't lose any more genetic variation. Right. So there's two ways that populations tend to get small so that this genetic drift is going to be a problem. And so genetic drift is only a problem in small populations. So genetic drift is going to be a problem when a population gets small because it went through some kind of catastrophe. Right? So if we have an organism just through bad luck, some climate change happens, some volcanic eruption, some hurricane blows through and kills off a lot of the individuals. And it just happens at random, right? It's not killing those individuals because they had worse adaptations to their environment. They were just in the wrong place at the wrong time. The original population is almost always going to have a lot more genetic diversity than those few surviving individuals. This is called a bottlenecking effect. The reason why is we shook the bottle and only a few individuals came out. All the beads or chips that are left inside the bottle represent all that genetic diversity that's lost. Right. So this is how we can go from having high genetic diversity to low genetic diversity. And so those frequencies of those alleles are going to change. That yellow allele is gone now. Its frequency went from some finite proportion to 0. Right, so genetic drift can happen through a population bottleneck. A lot of our endangered species have had this happen because they were hunted nearly to extinction, and so their populations decreased very rapidly. Another way that a population can have genetic drift happen to it is if it doesn't get reduced from a large population to a small population by mortality. But I'm going to draw this on the board. If we start out with a geographic area that's large and has a lot of individuals in it, I'm trying to draw some individuals here. Lots of individuals, lots of, not letting me draw. There we go. My large population with lots of individuals. And something happens so that a few of them get transported to a new location. So the most often scenario where we see this happening is that we have a small number of birds, say, they get stuck in a big storm, they get blown out over the ocean, and they are lucky enough to find some tiny island and they land on it and they survive. So those few individuals are going to be the founders of a new population in that environment. The exact same thing happens as in a population bottleneck. It's going to be a small number of individuals. They are not going to reflect the same proportions of all the alleles that the original population did. And so we call this circumstance a founder's effect. And there's some really interesting examples of this. There's island in the tropical Pacific where all the inhabitants are colorblind, which is very unusual because in most populations, it's only males that are colorblind and it's a small proportion. But in this case, the population that came to that island happened to have an unusually high proportion of colorblindness, and it ended up becoming fixed in the population. All right, so we can have genetic drift happen, whether we have a bottleneck event, whether we have a founder's effect, or if we have a small population for whatever reason is going to tend to have, so we have a genetic drift happening during this bottlenecking event, during the founding event, and then we'll actually tend to see subsequent genetic drift because it's a small population and those allele frequencies are going to continue to change once the population starts to reproduce. All right, so that's genetic drift. Oops, not ready for that one yet. There's three more forces of evolution. We already talked about natural selection. So I'm going to talk about the other two fairly quickly. One of them is mutation. Mutation, let me go back up to the, mutation is going to be a change in a gene. All right, so this could be a point mutation. There's a mutation that creates a new allele. Now we have a new allele in the population that wasn't there before. It went from 0.00 probability to having some finite probability. Mutations may be a benefit to the population. If they're useful, they may be a detriment. So mutations may be positive or negative. Over the evolutionary history of an organism, all of the genetic diversity that is present is due to there having been some mutation in the past. The fact that we have different eye colors in the human population means that there were mutations that caused those different colors to happen. All those different lap alleles that blue mussels have, all of them from 1 through 94. Some point in the population, there weren't that many, there were less. There were mutations that occurred that invented those new versions of those alleles. The last of these forces that can cause a change in allele frequency is migration. So notice how I have my diagram here with my populations moving back and forth. So every time there's an individual moving from one population to another, it's a migrant. Right? We use that word to talk about people who are coming into or out of the United States or leaving their home countries. It's the same idea, right? Movement of individuals from one population to another. Now, our populations aren't divided from one another in quite the same way we're talking about in natural species. Humans are kind of spread real thin across the whole planet. We don't tend to be separated from one another in quite the same way. Now, we used to be. It used to be very infrequent. If we go back 5,000 years, it would be very infrequent for individuals from the different continents to meet one another, even from individuals in, say, North and South America to meet one another. Now we move around very rapidly and far. All right, but suppose we have our caribou population that we looked at before, those two herds that are next to one another. So a young male might move from one herd to the other. That would be migration. All right, so he would be an emigrant, a leaver from one herd and an immigrant into the other herd. So migration has the power to change allele frequencies depending on how many individuals move and what the proportion of their alleles are compared to the population they're moving to and that they're moving from. Right, so suppose there's an allele that's missing in population B, but population A has it. Right, if one individual with that allele moves from A to B, then it's almost like there was a mutation, right? We've brought a new piece of genetic information into that population. So migration is also known as gene flow. Right, we tend to think of migration as being like I can observe an individual moving. Right, gene flow is thinking of not that person as a person, but as a packet of genetic information. Right, that packet of genetic information is moving from one place to another. I am more than my genome, I like to think, at least. All right. So those are the fundamentals of population genetics. Now we're going to turn our attention to species. We're going to start by talking about how species stay separate from one another, because understanding that is important in understanding how they are formed in the first place. One of the examples we're really familiar with is a bird's singing. So the male bird gets up there and he sings his song. Insect gets up there and he sings his song. A male human being might be standing or sitting somewhere and say their song, right? The reason the species has a specific vocalization pattern is that the female will recognize that as being the right kind and be sexually attracted to that individual, right? So the males and females can tell that they are of the same species, right? There are not only songs, there are also dances. And so we're going to call these ways that organisms can make sure they're mating with an individual of the correct species, of the same kind as themselves. We are going to call them species isolating mechanisms, right? We want species A and species B to stay separate from one another, right? So we want them to stay isolated. That's a good thing. All right, we're going to divide our species isolating mechanisms into pre-zygotic and post-zygotic, right? What's the zygote? The zygote is the fertilized egg, right? So pre-zygotic barriers are going to try and prevent mating from happening. They're going to try and prevent sperm and egg from fusing. Okay, remember we talked about in fertilization the sperm binding receptors on the surface of the egg, right? The sperm and the sperm binding receptors have to match. So that is an important pre-zygotic barrier to hybridization, right? Or to mating between species, individuals of different species, right? Now, suppose the worst case happens, the male and the female do want to and physically manage to mate with one another and sperm fuses with egg, then there's going to be some negative consequences. So these negative consequences that happen as a result of the genomes not fitting together quite right, we're going to call those post-zygotic species isolating mechanisms. So the pre-zygotic barriers are really trying to prevent hybridization from happening in the first place. The post-zygotic barriers are really the negative consequences or the bad things that happen as a result of the sperm and egg from two different species fusing. So things like those hybrids have lower survival. That's an example of a post-zygotic species isolating mechanism. It's a little different, thinking about it a little differently. There's sort of things trying to prevent and then the consequences if it does happen. All right, so we can put these into several different categories. We find species that are physically isolated from one another because they use the habitat in different ways, or they use the habitat at different times. So we can have, say, two closely related snake species. One's terrestrial, one's aquatic. They never bump into one another. They're never going to get together and mate. Same thing with our skunks. One is diurnal, that is, it's awake during the day. The other one is nocturnal, it's only awake during the night. They're never going to bump into one another unless one of them has insomnia. And so they're isolated from one another because of their behavior. The mating rituals, the songs, the dances, however you want to describe them. Here are some birds that are doing a very complicated dancing ritual, blue-footed boobies. That's an example. Nature shows love showing these. So any kind of mating ritual is going to establish that the two species are, the two individuals are of the same species. Some of these behaviors are innate, that the individuals know how to do them from the time they're born. We don't know how that works. Some of them are learned from, or they're partially at least learned. We know now that birds' songs at least are partially learned from their fathers or from other males that are singing nearby when they're in their nest. Individuals may need to practice those behaviors for a while before they get it right and actually find a mate. All right, so if you're in the right place, you're in the right time, you've done the right song and dance, and you say, ooh, that looks like the right kind of individual that I want to mate with, a mating attempt may ensue. All right, so it's possible that closely related species may have what's called a mechanical isolating a barrier, and that is they physically have different body structures that don't allow them to mate. They may want to, maybe you're feeling it, but when they go and they try to get the copulatory structures together, they can't do it, or the structures don't fit, right? Because if we have internal fertilization, we have a penis, we have a vagina, they have to match up in size and shape, right? So these are called mechanical isolating mechanisms. They're very fun to talk about. This example is two snails. You notice that they're coiled clockwise and then counterclockwise. Those are two closely related species. They can't mate with one another. Is it important that their shells coil the opposite way? Maybe not incredibly, but that that mutation happened. They're snails with one way coils and the other way coil, and now they are two different species because they cannot mate with one another. All right, so gametic isolation. Gametic isolation is that one that we already talked about. The sperm and the egg won't match together. Lock and key doesn't work. And so the fertilization isn't going to happen. This is really important in environments where, like marine and aquatic environments, where sperm and egg are released into the environment. There's no song, there's no dance. You don't even know if your neighbor is the same species as you. So those sort of free swimming sperm and egg really need to be able to match together on their own. Some closely related species, if you put their sperm and egg together in a petri dish, they may fuse, but they may never get together out in nature, so it's kind of irrelevant. All right. So suppose none of these methods worked, fertilization happens. So basically, the offspring can either have a reduced survival or reduced fertility, able to have offspring, or the offspring are okay, but their offspring have reduced viability or fertility. Right? So being viable means being alive. Reduced hybrid viability means that if there's a hybrid offspring, it has a lower survival rate for some reason. It may not be very well matched with the environment. Its body processes may not work quite as well as a true breeding species individual does. So it may have a lower survival rate. Reduced hybrid fertility means the hybrid offspring. So for example, the mating between a donkey and a horse makes a mule. Figure K is the mule. Mules are sterile. The reason for that is that donkeys and horses have a different number of chromosomes. And so when those fuse together, they can form an offspring. But then when that offspring tries to go through meiosis, it doesn't have the right number of chromosomes for any sperm or egg to come out normally. There could be reduced fertility. There doesn't necessarily have to be sterility. Hybrid breakdown is kind of a catch-all term for, so we have a hybrid, one parent was of one species, one parent was of another one. It tries to mate. Who does it mate with? Does it mate with another hybrid? Does it back cross or mate with one of the same species that was its mom or the same species that was its dad? In any event, its offspring either don't occur or have a lower survival or fertility. And so hybrid breakdown is basically, we got tired of listing things that could happen and anything else that happens to the grandchildren, we're going to call it hybrid breakdown. All right, so if we haven't prevented the hybridization from happening, there may be these negative consequences. Now that will act to, over the long run, it'll be a negative selective pressure against the hybridization, right? So there's a reduced survival, a reduced fertility. And so that behavior of wanting to breed with an individual that's not of the right species, it should be selected against by natural selection. Now, sometimes there are species that are closely related. They don't seem to have any isolating mechanisms, that they have hybrid offspring, and they seem to be fine, right? So again, we've decided as humans, as scientists, that we may want species to be different than one another, but it turns out that the species may not care so much as we do. And more recent research has actually shown that there have been these kinds of hybridization events that have occurred at least somewhat routinely across the histories of a lot of different species. So this, when it happens, we have a hybrid somewhere back in the ancestry of a species. We call it introgression. So introgression is basically, we have genetic information from another species that we find in our species. It may be a really small part, but it's kind of a leftover remnant of the fact that this hybridization happened. In humans, we have some Neanderthal DNA. A lot of Northern European people have about 3% of Neanderthal sequence that's detectable in their DNA. So that's an introgression event as well. So this example is from the big cats, and it turns out that there have been these events of crossbreeding between multiple lineages. So we used to think as scientists this didn't happen very often. As we look into it more carefully, we're finding it in more places. All right, our last topic of this lecture, which I know is getting kind of long, is how do we form a new species in the first place? All right, we have to have some kind of separation between the individuals. They need to be in populations, not all mixed together in one population. There has to be some reason that they don't mate with one another, and then they evolve characteristics such that they have formal species isolating mechanisms. So they have to evolve a species isolating mechanism, and then they are two different species from one another. It turns out there's two different ways this can happen, and they have to do with whether there's a physical separation of the two populations and then the species isolating mechanism happens, or do we have them actually geographically mixed together and they somehow manage to develop an isolating mechanism even though they aren't physically separated from one another, right? So whether the species isolating mechanism happens when they're mixed together or after they're physically separated is the thing that's different between them. So if they're physically separated, geographically separated, we call that allopatric speciation. So in this example, we have a lake that has all these red fish in it. If the thing happens on the left, that is, there's a landslide and it fills up the middle of the lake and it separates the lake into two lakes. Now the fish can't get together. So they just continue to evolve, and eventually they may have different enough characteristics. For example, one of them turns blue, that if we put them back together again, they wouldn't successfully mate with one another. So we find a lot of places where we have some kind of geographic separation. For example, if we look at at Central America and we find there's pairs of species that are very similar to one another that occur on the Caribbean side and on the Pacific side. So when the sea level was previously higher, those were one population, they mixed freely. Then sea level decreased. Now there's a land bridge separating them, the Isthmus of Panama, and we find that they have speciated. The other thing that can happen is that we have one lake and that somehow the fish develop differences. They have genetic variation, right? Then they start doing that non-random mating, right? That there may be some genetic variation where some of the fish have an allele that means they turn blue, and that's just part of the normal genetic variation. But individuals, blue ones only will mate with blue ones, and the red ones will only mate with the red ones. So if they change their behavior so that they have this non-random mating, then they can eventually diverge enough to be different species, right? So they developed that species isolating mechanism first, right, while they were still geographically mixed together. That is called sympatric speciation. All right, once some divergence starts to occur, there's three possibilities that can happen. So this diagram is showing that we start with 4 populations here. The circles represent populations. The little arrows show that there's migration or gene flow happening in between them a little bit, right? So the individuals within the populations are 100% well mixed. The populations are mixing a little bit with one another. Now, suppose that gene flow to one of those populations stops, right? So we see this red line, this barrier to gene flow. These other populations are still mixing with one another, but again, say there's an earthquake, volcano, or landslide, or change in sea level, and this population now has no mixing with the others. Initially, it will be very similar to them, but as time passes, natural selection will occur, evolution will happen, and because these populations are separate from one another, it's going to happen independently, right? Especially if the characteristics of this environment that this population is in has changed or changes. So as time passes, now this is representing, so this is like a evolutionary tree diagram on its side. So we're showing the potential of a node, a speciation event. So they start to diverge from one another in characteristics, and the changing color from blue to pink is representing that characteristics have started to change. Now it's possible that it becomes possible in the future for these individuals to mix with one another again, right? So the land bridge changes, the barrier is broken up, the populations move back together again. All right, so we have this potential for this population to become a new species compared to this other one. Right? So this is like species 1 in my original diagram, and this is like species 2. It's possible to have an incipient speciation event, one that's starting, all right, but not end up happening, okay? So if they start to diverge, but they don't diverge enough to develop an isolating mechanism, and then they're able to mix back together with the other populations, then they won't have turned into a new species. And so that's shown on the right-hand side here, fusion. That is, once they're able to mix again, they simply merge back together again, become mixed, and are still one species, right? So they almost got to be 2 different species, but then they didn't. Reinforcement of the separation is going to be what happens if they have become different enough that they continue on their trajectory to become 2 new species, 2 different species from one another. So reinforcement is going to be that the speciation event gets reinforced, that the one population that diverged is different enough that there is now a species isolating mechanism and they aren't going to mix together. All right, so then we get the 2 species where there was 1. Now we have discovered, maybe over the last 50 years, that nature doesn't do what we want it to, and that sometimes we find that species have diverged, or populations have diverged, and there is a certain distance between them, and there's some hybridization, and it keeps happening, and it's at a predictable proportion, and that they keep doing this. Right, so we call these stable hybrid zones. And basically those two branches of the tree diagram split apart, and then they stopped splitting, they became parallel. Right, so they're sort of stuck in this intermediate, they haven't totally split, but they haven't totally stayed together again. My personal understanding of what's happening in a stable hybrid zone is that they're still in the process. We've caught them, we're looking at them, and that somewhere down the road in the future, either reinforcement or fusion is going to happen, depending on what happens with the environment. All right. So, whether we're zoomed in or we're zoomed out on this process, it's going to look slightly different. So I think these stable hybrid zones, we're zoomed in, and we'll have to come back in 1,000 years and see what happened for us to know what's going on. Because remember, evolution can happen over short time frames, but it also can take a very long time to happen. All right, and our very last item to do with how speciation happens. It was a debate in the scientific literature for a long time about whether speciation events would happen quickly or they would happen over a long time. The diagram on the top is showing what scientists were thinking would happen if speciation happened quickly. So if we want to say, wow, a new species arose, then we have some punctuation at the end of that wow, we have an exclamation point. So the punctuated pattern is a way of saying we got big differences between our species rapidly. The gradual pattern is saying that our two new species started out fairly similar to one another, but as time passed, they slowly got more and more different. So after quite a long period of discussion in the literature, scientific community has landed on all of the above. That sometimes speciation events can happen. There's a change in an allele, it causes a big difference somehow, and that it persists over a long period of time. Sometimes speciation events happen gradually. It also depends again on what time scale we're looking at. If we're looking at the fossil record and we're missing some fossils, it's going to look like a punctuated pattern. But maybe as we find more and more fossils, then we may be able to see that there was gradual change over a long period of time. All right, so these are just a few things to think about when we're thinking about evolution and about how it happens and how it can create new species, and the relationship between how the process of evolution happening with these different ways it can happen, how that contributes to changing characteristics, particularly species isolating mechanisms. So as you think back over a couple different lectures we've had about evolution, I'd like you to kind of rethink sort of what we talked about originally and think about how these different concepts fit together, because they are all deeply intertwined with one another. And you'll have a better understanding of all of the concepts that have to do with evolution now that we have talked about these different topics. All right, thanks for listening today.